Man. Yeah, good.
Yeah, we're trying. Yeah, I've resent him the details. So
fantastic. So can I just give you a huge warm welcome, the gang. This is our biggest Coffee Buddies I think we've had, and it deserves it because we've got fab special guests. Just by way of introduction, this is Coffee Buddies. It's a get together. We're a community. We're all about supporting each other, and it's for the length of time it takes us to drink a hot cup of coffee, especially if it's got a badger. And today's special guest, which I'm more than pleased to have, is Michael Watson. Mike is a great guy. I've got to know him and worked with him recently, and he has the title of a vaccinologist, which I think is a very, very interesting title. And he's really at the cutting edge of some of the things that are going on with COVID and other things. So we thought we'd get Mike. So Mike, welcome to Coffee Buddies.
Thanks, Tony. It's great to be here.
Yeah, so Mike, just by way of introduction, tell us who, you've got a fascinating background, but please tell us who you are and what the heck is a vaccinologist?
Yeah, so I'm a Brit. I'm a UK physician. I spent 5 years in the glorious NHS and then realised I needed to go elsewhere, and I spent the last sort of 25 odd years in the industry, most of that in vaccines. And I've been fortunate enough to have worked UK, France, US a couple of times on a whole range of vaccines, Gardasil, pediatric combination vaccines, dengue. And then most recently, I've spent almost 4 years in the US leading Moderna's vaccine infectious diseases group, which was called Valera at the time. So that's mRNA. That's one cutting edge bit. The other bit that I'm focusing on these days is molecular design of vaccines. So trying to drag vaccines from the sort of breeding approach really that was used historically to something much more based on characterization and molecular design of antigens.
Fantastic. And you've got some slides to share with us, Mike, on your thoughts on COVID and some of your opinions. So, I mean, from a perspective of someone who's been with Moderna, this is very interesting. So if you can bring up your slides. Here we go, Mike. We're getting them up. We're getting slide number 1. If you can press it on, that's it. We're off.
How's that?
Yeah, lovely. So perhaps you take us through some slides and we'll chat as we go along.
Brilliant. So what I try, I'm assuming, and I know you're a very smart bunch of people. I know you're all keeping up to date with everything that's going on. So what I don't want to do is tell you stuff you already know. What I've tried to do is take a step back and look at this whole situation, see what we can learn from the origins of the coronavirus, see what the epidemiology is telling us, where we are in terms of treatment prevention options, and then I'm sure we'll have lots to discuss. There on the first slide, you can see the genome of SARS-CoV, and you can see the all-important spike protein. That's the S protein, the S1, S2 protein from the side or turn so you can look down on it. And that's what binds onto the ACE2 receptor and allows the virus to enter into our cells. And the ACE2 receptors you find most prevalent in the lungs, in the GI tract, and in the kidneys, which is why you're reading about respiratory disease, about some intestinal symptoms, and about a lot of renal failure.
Okay, fine.
What's important to know, though, is coronaviruses didn't just jump off a passing comment. Actually, they've been around a long time, and those that look at genetic evolution and wind the genetic clock back tell us that the coronaviruses have probably co-evolved with all the so-called amniotes. That's anything that has 4 limbs and gives birth through amniotic sacs. And so that's the 325 million year history. And so down at the bottom, the coronaviruses that we're really interested in are the mammalian coronaviruses, so the alpha and the beta. And those are the ones that infect an awful lot of mammals, including us. And then the delta and the gamma coronaviruses that infect an awful lot of birds. Now, coronaviruses have been around a long time. They're circular. You've got a nice picture of one here, and they have these club-like projections, which we call spike proteins, but you can see they look more club-like, and that's where the coronavirus, the crown name, comes from. They're big viruses physically, but they're also very big viruses genetically. Most RNA viruses will be of the order of 8 to 10 kilobases. And as you know, RNA relies on its low fidelity replication for its survival. Because it's low fidelity, it's continually mutating, it's continually changing and evolving. If you had a 30 kb RNA that was doing that, you would probably mutate yourself out of existence. So the coronaviruses rather uniquely have an RNA proofreader, something you most normally find in DNA organisms. And that means that actually, even though they are RNA viruses and they do mutate, they do so at a rate that is less rapid than one would expect for an RNA virus of that size. We've known about coronaviruses since the 60s. As you know, they've been around for millions of years. And we've had OC43, which has been around since 62, and 229E, which has been around since 67. We then, as you all know, in 2003, SARS-CoV appeared. And at that point, people went off looking and said, well, are there other coronaviruses out there? And they very quickly found one in the Netherlands, NL63 in 2004, and another one in Hong Kong, HKU1. We've since had MERS outbreak in 2012, and today we have SARS-CoV-2. So the viral name for COVID-19 is actually SARS-CoV-2 because it's most closely related to the beta coronavirus SARS-CoV. And what's interesting is as you look down the side there, the animal origin is bats or rats and mice. And given that rats and mice are essentially, if you put wings on them, you'd have bats. This is a rodent-derived infection. It does have intermediate hosts, camels for at least 2 of them, and ungulates for OC43, and potentially civet cats. And there's some discussion about what the intermediate host is for SARS-CoV-2. What's really interesting, though, actually, is if you do that same retrogenetic analysis and you plot back OC43 to when it first emerged in man, the line, fairly reliable, comes down to 1890. And in 1890, there was a pandemic. It was called the Asian influenza pandemic. But interestingly, the influenza strain that caused that has never been identified. There were 1 million deaths worldwide at a time when the global population was 2 billion, rather than today's nearly 8 billion. And it was somewhat unusual for influenza in that there were some neurological symptoms. And what's really fascinating, if you look at this cartoon from 1890, if you look at the top, you can see the patient, but look down in the bottom corner, you can see a figure with la quinine written on her skirt. And you may know that chloroquine and hydroxychloroquine are derivatives of quinine. So some things haven't changed.
My goodness.
So we're still learning very rapidly about how COVID-19 behaves, but we can look at the other coronaviruses to guess at how it might behave. So the other coronaviruses show a seasonal pattern. They probably cause somewhere from 4 to 15% of acute respiratory disease, and they may cause as much as 30% of respiratory disease in peak periods. And what's interesting is for cohorts where you have pre-existing antibody titers, we can see that actually infection happens in about 80% of people who already have existing antibodies. The message is that for the coronaviruses that we know and that we have good data on, being infected with a coronavirus doesn't protect you from later infection. And that's entirely compatible with what we see for other respiratory diseases such as RSV, human metapneumovirus, PIV3.
Just why doesn't it?
Why doesn't it?
Why doesn't
Great
it?
question. It may be to do with the route of infection. It may be to do with what I'll show you in a minute, which is the decay in the antibodies. So the ability of the virus as a survival advantage to induce a short-lived immune response so it can keep infecting people. So if you can think of a survival strategy, influenza's survival strategy is to keep changing. But it looks like some respiratory viruses, their survival strategy is just to make sure that they don't induce a strong immune response so they can keep coming back.
Right.
And the other interesting epidemiological feature there. If you look in the bottom right, assuming your pictures aren't across it, that the percentage of samples that are positive for OC43 in people with severe respiratory disease is fairly consistent through the age groups until you get to the 80-year-olds, and then it starts to go up. And again, that's very much, as you know, what we're seeing with COVID-19. So here's the immunology data I was talking about, and there are a couple of studies here. The first is not so very long ago, we were allowed to infect people with viruses. There was the Common Cold Centre in the UK, the MRC's Common Cold Centre, was busy infecting people to see what happened. And back in the late 80s, they infected them with 229E. So that's an alpha corona. It's not a beta corona, but it's a coronavirus. What you can see is that the dark dots are those volunteers that were infected, and the unfilled circles are those are the controls. And you can see the antibody titers go up, but by a year later, they've come right back down to the level of those that weren't infected. They haven't come back down to baseline, but they've come back down a very long way. In that same study, they then reinfected those individuals. So first time they challenged them, all 5 of the challenged individuals became infected. And one of those 5 was noticeably unwell. When they re-challenged them a year later, 6 out of the 9, so they re-challenged the controls as well, became reinfected, but none of them became unwell, and the virus shedding was shorter. So they were able to reinfect them a year later, but there was evidence that there was still some residual protection based on the fact that the virus shedding was reduced. The curve on the right looks then at a cohort of 23 individuals who had SARS back in 2003. And what you can see from the black line is that over a period of 5 to 6 years, their antibody titers, so these are anti-SARS IgG, they declined right back down below the detectable level. The green line is one of the patients whose antibodies didn't change at all, and the red line is another patient whose decay was slower. So the message here is that overall antibody titers do disappear, but it's not the same for everybody.
But just one question, right? I mean, is an antibody measurement seems a rather antiquated measure of what we now know is a complex immune system.
Yeah.
And, you know, it's almost Jenner type of era measurement. I mean, if we put a more sophisticated measure of immunity in, I don't know, something to do with T cells or whatever, would that, I'm just intrigued whether this is the right measure for immunity.
That's a great question, Tony. And what you can see below there is that whilst all the IgG disappeared, if you looked for the B cells, they also seem to have disappeared. But what was still present in 60% of the individuals were memory T cells. So you're right. We have to be very careful about assuming that it's just detectable IgG that's important. And the T cells were still there in 60%.
Yeah.
That also may be an indicator of why the elderly are getting more sick, in that, as we know, T cells decline and function less well in the elderly. And it may be if that had been an elderly population, that would have been even lower, and that may be what's going on. That's one of the possibilities.
Interesting.
So the next thing I want to touch on is, and you may have seen these curves, but what's been driving decision making? And these curves come from the modeling group at Imperial, and you will have heard Neil Ferguson's name being mentioned frequently. And on the left, you can see really what got people scared. And that is that if we had left the epidemic to unfold as it was unfolding, you can see the line starting to come up there from April, that their projections suggest that by middle of June, we would have had 500,000 deaths in the UK and over 2 million deaths a little later in the US. So you can see why everybody reacted so strongly at that point, and why we hope we're now turning at about the line where that 5,000 deaths per day,
and
5 deaths per day, 100,000.
And Mike, what did Neil have that others didn't, because the prediction before wasn't what was produced. You know, what was his twist that all of a sudden seemed to come left field?
That gave such scary numbers.
Yeah.
What did he have? I mean, he had a lot of practice at doing this, and he's done this a lot in the past with diseases, and what has happened has been very close to what they predicted. And then I think what they got was data coming out of Wuhan to allow them to monitor that. And most importantly, they had data that allowed them to estimate the R0, the basic reproduction number, which is for most models the critical number. You can model without it, but for most models the critical number. So I think that's what he had, was an R0.
Yeah.
They then wanted to look at a range of different mitigations, and that's the graph on the right. And importantly, you can see a red line along the bottom there, and that red line at the bottom was the estimated intensive care capacity of the UK. Hence the fear that we were going to just totally outstrip that. And what you can see is the black line is essentially the line you see on the left there. Doing nothing meant that wouldn't change. The green line is schools, and you can see that's why they were less urgent with schools, because that had the least impact. You then see case isolation and quarantine, and you can see the blue line there, the most impactful policy is essentially what's put in place. Now, the challenge, of course, is that we've got the tiger by the tail. Whilst you've got a tiger by the tail, as long as you hold it high enough, it's not going to bite you. But the moment you let go, it's going to turn around and bite you. So the moment we start to let go, what happens? And those same models suggest that we could see a second piece, or we should expect to see a second peak into the autumn and the winter. So there are two implications here. One is, what do we do between now and then? And the second, of course, is that we've got this critical window when we need to get vaccines and therapeutics developed and made so that we can intervene as that second peak comes. And classically, pandemics behave exactly like this. They have first peaks, they have second peaks, and they have subsequent peaks.
And that's a very narrow window, Mike.
It is a very narrow window, absolutely, but it's a window. And I think it's focusing minds on the fact that we have to get something done this year. Yeah. Influenza pandemics, often we talk about annual peaks, so you might have longer. And then these are data again from Ferguson's group. What they've done is they've taken the Wuhan data, but they've adjusted it to try and adjust for asymptomatic cases, for a bunch of other potentially unknowns. And what you can see here is the estimated infection fatality rate. So what is the risk of dying if you're infected with COVID-19? And on the one hand, you can see what we're all seeing is that the elderly are particularly at risk. There's close to a 1 in 10 chance. Actually, down at the lower age groups, the risk is extremely low, and it's 0.06% in most of the young age groups. So what can we do about it? What are we doing about it? There are really 3 main points of intervention. The first is primary prevention. That's the equivalent to stopping someone getting blood pressure. Secondary prevention or primary treatment, that's the equivalent to making sure that someone who has high blood pressure doesn't have a heart attack. And secondary treatment is the equivalent of treating someone when they've had that heart attack. So in terms of primary prevention, the social interventions which are designed to get that R0 to less than 1. Of course, vaccines are ideally what we want. And those vaccines fall into 3 broad groups: the nucleic acid antigens, so the mRNA, DNA, protein vectored antigens. So it's the antigen delivered either attached to a virus, such as the Oxford group is doing, or just adjuvanted and delivered with an adjuvant, or a whole virus where you may take a whole virus and inactivate it either genetically or chemically and then give that to the individual. For those particularly at risk, if we have drugs that work, you may consider giving them the drugs prophylactically. And then in terms of secondary prevention and primary treatment, you will have all read about remdesivir. That is remdesivir above there. What you can see, that's a nucleoside analog. So that, that tricks the body into thinking it's a nucleoside and gets incorporated into the virus, and the virus therefore can't survive. You may have seen news about remdesivir, and I'll show you those top-line data in a couple of slides' time. People are also looking at whether convalescent serum, so serum from people who've recovered, whether that helps to treat disease, whether we can make antibodies in horses and camels, which is what was done back in the early 1900s, or whether there are recombinant or mRNA approaches to antibodies. And there are others. You've heard the chloroquine, hydroxychloroquine story. The jury is still very much out on that, although increasingly it suggests that chloroquine and hydroxychloroquine are not helpful, and in fact may be associated with increased risk. And then secondary treatment, you again have probably all heard about the cytokine storm. This cascade, this rapid uncontrolled cascade of cytokines organized by IL-6, which gives rise to disseminated intravascular coagulation, DIC, shock, lung injury, cell death, renal failure. There's supportive treatment, hospitalization, ventilation. There are people looking at specific immunomodulation. Can we block IL-6? Can we do anything else to modulate those cytokines and chemokines? And lastly, many are using steroids as they did for SARS. If you just suppress the immune system, what impact does that have on outcome? And this just being a vaccinologist, this is the vaccine view. That number at the top is out of date. There are, at last count, over 125 vaccine candidates out there, which in itself is interesting that so many vaccines can appear just like that. 8 or 9 of them are already in the clinic. We expect about 25 of them to be in the clinic by the end of this year. And just breaking them into those 3 groups that I talked about earlier, the most rapid are the pure spike protein antigens that you saw right at the very beginning. There are at least 10 mRNA versions of that, at least 10 DNA versions of that, and at least 25 adjuvanted protein versions of that out there. Vectored antigen, that's the approach that the Jenner Institute, the Vaccitech guys are taking, using an adenovirus vectored spike protein. And there are a large number of those out there. And then whole virus, there are a few of those out there, but there's concern that might come with a risk associated with something called enhanced disease. And we can talk more about that if that's of interest to anybody. And then I put this in really, we talk a lot about antivirals, and antivirals are important. But as you'll see from a minute for the data from remdesivir, once somebody is really sick, antivirals often have a very limited benefit. It's one of the reasons we see them used as sparingly as they are in influenza, despite having influenza antivirals. The other is, if you want to use them earlier in the process, you've got to have an incredibly good system for tracking your virus. So what you see on these 4 curves is, for all of them, the vertical axis is the simulated outbreaks controlled. So if you want to control 100% or 80 to 100% of them, what have you got to do? And what you can see along the bottom axis is that you've got to trace a lot of contacts. You've got to have relatively few initial cases. You've got to know that you've isolated people very shortly after onset. So in short, what this data tells us is that yes, antivirals are important, but you've got to have an incredibly good detection, isolation, and intervention system in place for them to really have an impact. So we should never be in a position of thinking antivirals or vaccines. It's definitely both, but at a population level, there's nothing that will replace vaccination or herd immunity if we all end up getting infected. So these are the hot off the press remdesivir data. This was a study finally in 1,000 subjects. There've been an awful lot of very small uncontrolled studies going on. People were infused IV daily for 10 days. And what they found was that remdesivir recipients got better 30% faster than placebo recipients. That's good. That means you're likely to spend on average 11 days being treated rather than 15 days in hospital if you're on placebo. In terms of mortality, the mortality could be lower on remdesivir. The numbers in this study weren't big enough. You'll see the p-value there is still above 0.05. But suggesting it does have an effect on mortality, but not an enormous effect. It's not halving mortality or saving lives completely. So just to summarize all of that, coronaviruses haven't just walked in to the shop. They've been around a long time. COVID-19 is in many ways behaving in the same way as other coronaviruses. The epidemiological data suggests that once established, as it is, SARS-CoV is not going to disappear. It's not going to disappear, A, because the R0 is too low for burnout. If you have a very high R0, you need a very big population to sustain it. But an R0 of 2 to 3 would allow it just to quietly keep burning through the population, just as flu does. We know that humans can be reservoirs, and we suspect that non-humans can be reservoirs as well, mammals. It's probably, not definitely, going to follow a pattern like OC43 and possibly 229E. And if it does, there's a very strong possibility that this is a virus that can reinfect us periodically.
So just Mike, before we move on to the last bit,
yeah.
I mean, what in a clinical, I'm interested in the design. I'm a small molecule guy, and so I'm interested in a vaccine. How's endpoint measured? It's quite straightforward in a world that I've always lived in, but I'm intrigued by the this idea of how you field test really the drug. I mean, so what? Just can you give us an idea? How does it work in terms of expansion of patients from normal volunteers out into a vaccine trial? Because all of us are interested in how long is it going to take. So we need an idea of the process.
Yeah. So normally, and of course, the development of vaccines, the risk benefit is very different from a small molecule because you're dealing with well people. So hence the process is often much slower. You need good tox data. Then you go into phase 1, and in phase 1, you will do sentinel groups. So is there any reason this is essentially a dangerous molecule? You'll expand up. And what you're just trying to establish there is, do I get an immune response with an acceptable local and systemic safety profile? If I do, then I can start to move in phase 2 towards establishing the final dose, the vaccination schedule, potentially in my target population. Now, that's okay if that target population is healthy adults, which it often is. But if that target population, as it often is infants or the elderly, you may have to do your phase 2 in your healthy population before now starting to down escalate into infants or up escalating into the elderly. When you've established your dose and your frequency of dosing in your target population, then you go for your efficacy study. And there are a number of approaches to efficacy. The standard efficacy involves usually very large studies where you're classically going to be vaccinating either 2-to-1 or 1-to-1 active to placebo. You're probably going to be looking at, you've got to look at least 5,000 subjects for a safety perspective, but you're probably going to be into the tens of thousands purely because of the incidence of many of these diseases. And then you follow them out. So there are some diseases that are so hard to do, you can do challenge studies, and that's really important in the context of coronavirus. And people are asking themselves, why don't we do a challenge study? Sorry.
So what are the tricks, Mike, to reduce it? So you know because what we have to do is take some educated risks here. So what's the tricks to compress? If the world needs to compress, what trick can we use?
So the reason that the RNA, the DNA, and the adenovectors are in the lead is that they've all done multiple phase 1 studies, multiple tox studies, and they've shown that intrinsically giving RNA, DNA, or an adenovector to someone is not dangerous. So if you like, chemically per se, you're not going to kill anybody by giving them this construct.
Right.
So that means that the tox, for many of those tox, has just been forgotten. And we said, look, we know this is safe. Let's just get into humans and see if it induces an immune response. Luckily, the target is, by and large, adults, healthy adults. So a phase 1 study, therefore, in many ways, is a phase 1/2 study. We can say, well, look, we know we're going to have to vaccinate people quickly. So if our vaccine doesn't work with 1 or 2 doses, with at most 1 month apart, it's not interesting. So we know what our schedule is. We know broadly what our doses are likely to be. Let's just get in. So the phase 1/2 studies that you'll see are just escalating through those doses to try and find a good dose.
Right.
Because because there's a suspicion that there may be a decline in antibodies here, we want to be on the higher side of immune response. So you'll see people opting probably for the higher doses. The next question is then, so that's why you're seeing the approach for Oxford and from others is let's do a phase 1/2 combined as a first study, and then let's get on into a phase 2/3 study. So rather than taking these traditional 3 steps, let's do it in 2 chunks.
But it still feels a long haul. And those of us who've been around know that these things always take they're a long haul.
There are. And I think now the next decision that regulators and policymakers and developers have got to take is, are we going to start using a vaccine based on immunogenicity alone? Or are we going to have to wait for full clinical efficacy data?
Yeah.
Right, Tony, we've got so many questions, as you quite imagine with this. So,
Mike, it's a Mike. Just can I tell you, awesome, buddy. Let's open it up for questions, Graham.
Okay, yeah.
Mike, can you bring your screen back?
Can you what?
Can you bring your screen back to non-slide?
Yeah, here we go. Yes, brilliant. Okay, we're going to have Pilar first, Sylvia, and then Tom.
Hi, Michael. Thanks so much for the presentation. My question is very simple. I'm just trying to figure out if there is a silver lining down there. So in the study from the 80s or 90s, I don't know if there was any investigation as to whether once you contract the virus, even though the antibody, the presence of antibodies in your body will diminish, does it mean that you react with in the same way when you contract the disease again, or will your body be a bit more reinforced for some reason?
Yeah, I don't think anybody looked at that as systematically as that, but the studies that were done, the EPI studies there showed that the coronaviruses were seen most frequently in 2 populations, one in student populations, where there were frequent reinfections, and no evidence though that anybody was getting sicker. But interestingly, care homes were absolutely where they saw the most impacted disease. And it looks very much like something like RSV. So whatever the immunological basis or the physiological basis of this, it seems like frequent challenge and boosting as a youngster is okay. But as soon as you get older, something is going out of kilter that means you start to lose that battle.
So it's gerophilic, basically, yeah. Okay.
Yeah.
Thank you.
Thank you.
Okay, if we have Sylvia next. Hi, Sylvia. You there?
Yeah, hi. Thanks. I really enjoyed actually. I was wondering a pretty simple question. From your perspective, which vaccine approach, mRNA, DNA, or so on, would you think would be the most successful? And the reason why I'm asking, actually, I work on DNA vaccination long time ago, 20 years ago, for tuberculosis. And it's really, really immunogenic, especially if delivered via gene gun and so on. But I'm not really up to date with the developments. I'm very curious to know your opinion.
Yeah, you know, it's like all these things, they all have their pluses and their minuses. DNA tends to induce lower IgG titers, but does tend to induce quite a good T-cell response. So if that's important, then DNA is not a baddie. The problem with DNA is the device that's required. So in terms of population-wide vaccination, it's not going to be the solution. It's good for targeted populations, but it's not going to be the solution population-wide. RNA seems to bring both, but at the end of the day, there are no licensed mRNA vaccines. So, you know, there are known unknowns there. Every indication from preceding studies, it'll give an immune response, but we'll have to see. The other challenge with mRNA is that it has to be frozen until the last bit of the delivery. So there may be some distribution challenges there. Then the vectored viruses look good. They may be one dose. They often give a good T-cell response. But then there are concerns about enhanced disease around vectored viruses because you're delivering a lot of other antigens. So if you're not inducing the right immune response, or you're inducing heterologous antibodies, then that can be concerning. So, you know, I think there are swings, roundabouts to all of those. Sorry about that. Swings and roundabouts to all of those. And I think hopefully all three will work, and they'll all be complementary and you know bring their own benefits. But we'll have to wait and see. Have I just?
Right.
No, that's good. There we go.
You're there. You're all good. And we have Tom next.
I think Tom's on mute still.
Tom, where are you?
Where are you? Here he is at the top.
There we go. Hi, Mike. Absolutely awesome talk. Thank you. A key differentiator between SARS-CoV and SARS-CoV-2 is the spike protein. Should we be overcautious with regard to our interpretation, with regard to the, concerning the viral infectivity of this? And should we be looking perhaps or evaluating more the non-structural proteins, you know, the proteases, etc.?
Yeah, I mean, it's again, pluses and minuses. I think when you look at serum from exposed animals, the vast majority of the neutralization comes from S protein targeted immunity. And there's always this debate about, therefore, you know, but if you go after a conserved antigen, well, it's conserved, so you might be able to cross-protect, but why is it conserved? Is it conserved because it's not under any immunological pressure and therefore isn't important immunologically? The first, and I think that the second is there are also these concerns about enhanced disease. If you target a non-neutralizing antigen, you can actually enhance disease because you actually facilitate Fc gamma receptor uptake into macrophages. So I think for those reasons, people have all but, I think that there are one or two companies out there looking at the nuclear protein. But other than that, people have shied away from that because it seems to be so clearly important to focus on the S protein. But you'd have to do the work and have a look at it. But it wouldn't be the first target to go after the non-spike protein. It seems so immunodominant.
Okay, thank you.
Okay, the next three are going to be Diane first, Suzanne, and then Kristin.
Hi, Mike. Thanks for your talk. I think my question's already been answered because it was, could you tell us a bit more about the risk with whole virus vaccines? Because one of our clients is one of the developers of a whole virus vaccine. So
Yeah.
it was like, how are we going to recognise that that's going to be a no-go? I mean, they're not in Europe at the moment. So, you know, we're not actually made any submissions, but how are we going to recognise that this is a no-go?
So you'll hear a lot of you'll hear a lot of talk about enhanced disease. That it goes back to 3 experiences: RSV and measles vaccines back in the 1960s, and dengue. And what happened with RSV is that these were formalin activated whole viruses, and the immune response that was induced was very Th2 skewed. So essentially characterized by eosinophils, neutrophils, macrophages, complement deposition and immune complexes. So essentially the vaccination primed them for allergy. So when they were re-exposed to the virus, they essentially had an extreme allergic response to that allergy. Now, that could have been because of the formalin activation, which produces carbonyl groups around both the RSV and the measles, or it could have been just because of antibodies being generated against non-neutralizing antibodies, or both. Basically, what you need to do is show in your animal model, A, that you're inducing a Th1 rather than a Th2 response, that you have a high ratio of neutralizing to binding antibodies, and that there's no evidence of heterologous antibody production. And also some structural work is useful, but that structural work is often harder with a whole virus. If you can show that, that immunophenotype in your animal models, then you're probably in a good place. But you may be asked to do animal studies in ferrets, in cotton rats, or in NHPs to confirm that you don't get enhancement. The last thing is to make sure the antibody titers are high, because the other thing that causes enhancement are low levels of antibodies, which enhance more than they neutralize.
Right. Suzanne, you've got a question for us?
Sorry. The focus has been very much on viruses and on antibodies. The issue of there being potential of reinfection, is that firstly going to be just a total challenge against us getting a vaccine? And is it secondly going to be against the government's idea that we can get out of this with immunity passports?
Yeah, I mean, of course, and I don't want to depress everybody with this question. At the very worst, and I think the important thing with all these scenarios is exactly that, is to look at different scenarios. So what's the worst? The very worst case scenario is we get to the end of this and discover that inducing high levels of antibodies against the spike protein doesn't help. I think that's unlikely. So the next question is, if it does work, and so the middle scenario is that it does, but you get limited immunity. And that's fine. You know, that's flu. We deal with that all the time. We vaccinate people every year. That's fine. That just means boosters. The other end is that this was all far too depressing. And the reality is that the vaccines are really good, and we get lifelong prevention. This is why you're hearing caution about antibodies. You're hearing the WHO saying, be careful about using passports until we know a bit more about all of this. So I think we need more data. We need more understanding before we know how helpful immunity passports will be, and what test we're going to need to establish immunity or not.
Right, that's fantastic. Kristin, are you?
There must be something. You know, if you've got your patient there and he gets better, because people do get better, something turns. You know, you can't. You're not. I mean, I know you can breathe and everything, but you know, there's something that changes. So I suppose the question is for the immunologist, what is changing during that acute period? There must be loads of people looking at that, surely.
Yeah, and I think if you look at the timeline of disease, those who don't go on to being very, very sick tend to get better in a time which is a very classic, innate immunity, adaptive immunity. T cells are coming on board. You're all good. Back home again. Those who then don't win that battle end up in ITU are there much longer, and then that recovery is all about tissue recovery and all of that. And some people argue it's as simple as viral load. If you got a big dose and you didn't handle it very well, then your immune system loses in that battle. It's 5 against 100, and if you do, then the immune system wins. So it's something about that balance, but it seems to be two very different kinetics. And the immune system is clearly working, and I'm not suggesting that worst case scenario is going to be the one. The question is then how long we're protected for, and we've got a precedent of respiratory viruses where protection is not lifelong.
Great. Okay, Kristin, you ready?
Hi. Thanks for giving this session, Michael. It's my question is a little bit. We can zoom out a little bit, just because it's not every day that our field is sort of on the front page of every single paper for months at a time. So you've got a lot of statements by a lot of people, that aren't always talking about this kind of stuff. And I just wanted to ask, what's your view on the comments sort of over the past week or so from Boris Johnson and Chris Whitty saying the only way out of this is through a vaccine?
I think there are, I think there were. I mean, there are 3 ways out of this. One is a vaccine. One is that everybody gets infected until herd immunity is up to 60 to 80%. Those are the 2 ways, really. And I think the third is that second, what I meant by saying is that actually it could be pretty messy if we allow that to happen. That's going to take 2 or 3 years when it's, it's going to be 2 or 3 pretty messy years. So I think they're right that we need a vaccine. We need something to cut this short. Otherwise, we're in for a very rough ride over 2 or 3 years as we go through these sequential peaks of this.
Okay, thanks, Kristin. Right, Panteli, Muz, Rebecca Todd, the next 3. Panteli.
Thank you. So Mike, thanks for the insights and the presentation. Highly informative. Quick question from me, just in the interest of time. So having been a stem cell transplanter, I understand the immunological basis of how do we develop, long-term immunity in these patients. And I just wanted to kind of get your thoughts on, is this going to be a sequential approach? Is it going to be a personalized approach when we think about the development of these vaccines, use of adjuvant, for example, even a cell therapy based approach, so there's a myriad of different approaches, but are we going to need to think about sequencing, and are we going to need to try and personalize it based upon your point? How do we develop, with a focus on the patient, how do we develop these long-term immune responders and really understand it from an immunological perspective? Thanks.
Yeah, I think and I hope this will reawaken the vaccine field in that a lot of the vaccines we have today are 50, 60, 70 years old, based on very old technology and based on no understanding of the immune system. And poor understanding of the host pathogen interaction and all the things you're talking about. I hope that we will now drag it all forward and ask ourselves all those questions and because I think we look at these, we're talking about whole virus vaccines. That's a very old technology.
Yeah. Okay. Thank you.
Right. Thanks for that question. Muz, are you there?
I am, yes, thank you.
And
thank you, Michael, for that talk. Just a quick one. So I think we've all heard numerous times about the 80% confidence of having a vaccine ready in September by the Oxford group. Bearing in mind all the steps that you talk through and what we need, what do you think we will have available as a regulatory package by September that will allow for approval?
I think in the best case, we will have efficacy data. We'll have vaccines into the right populations at the right time, and we will have efficacy data enough to give us enough confidence to start to roll the vaccine out more broadly. It's tricky though, because we're doing everything to stop the spread of the virus, and that's good for those who don't want to be infected. It's not good if you're trying to run a clinical study to show that your vaccine works. And so I think there's a second scenario where we haven't been able, for good reasons, haven't been able to establish that efficacy, in which case people will have to decide whether to make a decision based on immunogenicity and safety and say, look, we've given this vaccine to a couple of thousand people, everybody's well, there's no evidence of enhanced disease, we haven't got the efficacy data yet, we've got a great looking immune response, let's just crack on and use this under emergency use authorization. I hope it'll be one of those two scenarios.
Rebecca Todd, you've got a question?
Yeah, it was a related question, really, and it's a very simple one, which probably is far too simplistic. But you know, I mean, I'm sort of, and probably quite a lot of people are quite familiar with the sort of the data that's published. It gives you kind of an odds of a drug entering clinical trial of being effective, and I've got no concept of whether it's similar for vaccines, and whether the Oxford team being so optimistic has got any basis. And I was just interested to know, you said that 25, you expect 25 to go into the clinic this year. I mean, can we kind of give hope that one of them will come off, because that's, you know, odds on at least one will, or is, you know, do you just need way more than that, really? You know, what do vaccine success rates look like?
I mean, they're essentially all taking the same approach, which is to deliver the spike protein, usually in a pre-fusion anchored form, either as an antigen, either as mRNA, DNA encoding, or as that. So I think if one works, many will work. And then traditionally, if you look at the Tufts data on success rate, traditionally vaccine success rate is, vaccine failure in later stages is low, relatively low.
Oh, good. Mm-hmm.
There have been a few exceptions recently, but by and large, because once you know it's immunogenic and it's producing the immune response that you think you want it to produce, then you're usually off to the races. The big challenge for all of this then is going to be scale up. You know, 2 doses for almost 8 billion people is a lot of doses. So that's not going to happen overnight.
Reason for hope, though. Thank you.
Yeah, I think so.
So Tony, are we going to round up? I think.
My God, this has just been fab, Mike. So, I mean, you know, can I just give you a big round of applause from everyone, a virtual round of applause? That was super duper. I mean, perfect. I mean, eloquent and succinct, and we get it. I think we're all going to go away, those of us who have developed drugs for many years, thinking, my goodness, you know, these things always take longer than you think. And this time we've got to hope it's going to take shorter than we think. So can I thank you enormously for coming, and we'd look forward to seeing you again and bringing vaccinology to, I know you're in a very interesting new explosion of a company that you're going to build, and we look forward to hearing again about that, because I think we want to see vaccinology come back from the dark ages, and thank you so much. Graham, what have we got to look forward to the next Coffee Buddies?
Well, we're still working on it a little bit on Thursday, so hopefully we'll have some more news for you tomorrow on that. We might have a military historian coming on Friday for Motivation Buddies, and then on Monday the 11th, we have Kat Arney and Peter Penny on the psychology of the pitch and boring comms waste lives. So looking forward to all of that, and we'll keep you all posted.
So in the words of the great TV programs, be careful out there, and bye for now. See you next time. Thanks again.
Thanks everybody.
Bye.
Bye.